I'm Maria Wilson.
And I'm Danielle Mandikian.
And we are scientists. We love science.
Yeah, we do. So when we aren't doing it, the next best thing is to talk about science. And what's really awesome is that we're surrounded by some of the most brilliant minds in research.
We're going to step away from the labs today to talk to other scientists about the cool stuff they are thinking about, working on, and imagining.
As well as how some of these discoveries just might lead to new medicines. So grab your favorite drink, get ready to unlock your science brain, and join us for Two Scientists Walk Into a Bar.
The show for scientists, science geeks, and the people who love them. What do you think has been the biggest breakthrough idea in oncology research?
I think it's CAR T therapies.
Maybe using mRNA vaccines to target cancer is also really exciting.
I think it's PD-1. Checkpoint blockade.
CAR T?
I think maybe one of the biggest breakthroughs could be in cancer vaccines, but it's a hard space and it may not work alone. It's often in combination with existing standard of care. So excited to see what happens.
Hi listeners, I'm Maria Wilson, and today we'll be talking all about recent advances in oncology with our expert, Louis Vermeulen. Welcome to the show, Louis.
Yeah, thank you. It's great to be here.
So oncology has been a hot topic on this show. We've talked about the history and origins of immunology, mutations in breast cancer, cancer pathways, cancer vaccines. Where is oncology today?
Yeah, oncology is, I think, at a very exciting point in time. Of course, the advances in oncology research and the clinical implementation have been very, very fast in the last couple of decades, I would say. But certainly now, with all the new opportunities and new technologies that we have for diagnosing disease, studying disease, understanding its biology, but also to treat it, is, I think, great to be a part of it in this moment in time.
So my mom actually passed away from cancer. She had ovarian cancer, but she did have several very good years because there were new medicines being approved and coming to patients during that time she was fighting her disease. So I got to experience that firsthand, how important it is that a drug gets approved and gets made available to patients and used, because there really is a ticking clock when you have cancer, and that sense of urgency became very, very real for our family.
No, absolutely. And this urgency is something that, of course, you feel in the clinic, but also something that I feel also now every day, right? Because that's really what we try to do, is advance our scientific insights as fast as we can to make clinical impact, because the patients cannot wait.
I think one of the things we wanted to talk a little bit of is how early can we intervene in cancer? I know what happened with my mom is she was really quite late-stage disease at the time she was diagnosed, which makes it very difficult. She was never going to be cured, right? But could you talk a little bit about how we can get to patients earlier and potentially actually cure disease rather than just manage it?
Yeah, I think the earlier you find cancer in patients, the better you can treat it. And there are several reasons for that, I think. First of all is the simple reason that when it's localized disease, confined to one particular organ or part of an organ, you can use, of course, surgery in many cases. But there is also a molecular reason, and that is that when the disease advances further and spreads to other parts of the body and forming metastases, the heterogeneity of the disease increases at all levels. So genetically, the heterogeneity increases with, for example, different subclones that have different mutations, but also based on the microenvironment, for example, in different regions of the body that is different. And this variation at all those levels makes it more difficult to successfully treat disease that had advanced further. So I think there is a big advantage in trying to find patients that have early disease and treat those. But I think you can also take it one step further and maybe even prevent cancers or intercept very early lesions that are not cancer yet, because they are not invasive, but still have all the abilities to progress towards an invasive tumor. And those lesions are even genetically simpler, maybe, which makes them potentially more attractive targets for successful therapy and really curing patients and preventing them from getting cancer altogether.
It will be fantastic if all of these new diagnostic approaches pan out and we can really identify people much earlier in disease when the cancers will be much easier to treat. And of course, there have been a lot of really great breakthroughs in treating much later stage cancer and metastatic disease. Wouldn't you agree?
Yeah, I think the successes that we have seen there in some areas are no less than spectacular, right? When I just started my career in the colorectal cancer patients with microsatellite unstable disease, they had a very dismal prognosis when they had metastatic
What does that
disease.
mean? Microsatellite?
Microsatellite disease. Microsatellite unstable disease. That is a subgroup of the patients with colon cancer that have a lot of mutations in their genomes, and they are normally also more immune active. And if these patients present with distant metastasis, then they have. I had to say, a very limited life expectancy, because the therapies that we had at that moment in time, and mainly chemotherapy, were just not very active. However, with the development of immune checkpoint inhibition, for example, the prognosis of those patients is actually from being the lowest to having the best prognosis in that situation. And in some cases, patients are actually cured, even with metastatic disease. So that has been actually a tremendous advance.
These are patients who, because of the genetic instability, have a lot of antigens on the surface of the tumor. Is that the way to think about it?
That's probably part of the explanation, but also probably there is something to their microenvironment that makes them more susceptible to this type of treatment. And the real question is now, how are we able to expand on this great success in that relatively small subgroup of patients towards the whole group of colon cancer patients and beyond. And I think one of the things for the future there is really to understand better how the cancer cells interact with the microenvironment.
You're talking a lot about the microenvironment. Do you want to tell us exactly what that is and why it's important?
Yeah, so cancers do not only consist of cancer cells, the cells that harbor all the genetic aberrations, but they are actually complex tissues that contain immune cells, but also fibroblasts, blood vessels, neuronal cell types in some cases even, that also recently received quite a bit of attention. And it's really the interplay of all those cell types in the tumor microenvironment that dictates the biology of the disease, dictates the rate of progression, ability to form distant metastasis, but also the response to therapies.
It's almost like a new little organ that has been formed with its own sort of cellular population.
Yeah, absolutely. Cancers are in a way aberrant organs. Yeah.
We talked on this podcast just recently about cancer vaccine strategies. How are these other approaches different from a vaccine-based approach?
Yeah, a vaccine approach is something, of course, that is very interesting because it really enforces the activity of the immune system against particular antigens that are often expressed on tumor cells. And so you try to build a broader repertoire of anti-tumor immunity, while in the case of, for example, checkpoint inhibitors, you really try to put off the brakes of the existing immune response that is present. And probably there is a place for both of these approaches in specific, and maybe even combinations thereof in specific clinical contexts. And I think it's our job to try and find out what of these strategies is the best fit for which particular disease and what stage of disease.
Yeah, I guess I think I sometimes forget that it's actually one of the jobs of our immune system is to keep cancer under control, right? It's doing that all the time, actually, without us noticing, right?
Yeah, absolutely. And the point is, of course, that we only see it when this process fails. And so we also don't have very good estimates of how successful it is in most of the other cases. So it's one of the questions I have and I don't know the answer to. Is how often is the immune system actually very active in suppressing the outgrowth of a pre-malignant clone or even a tumor? I don't have the answer to that.
Maybe we will, as we get back to your earlier point about as if people have much earlier diagnosis, maybe sometimes the treatment is we're just going to watch this tiny little thing, and perhaps sometimes it just goes away on its own. Maybe, I don't know.
Yeah, who knows? Who knows? That would be great.
Yeah.
Hi, Maria.
Hi, Wellington.
Hi, Maria.
Hi, Karen.
So, I find this concept of early diagnosis and minimal invasive treatment really exciting, maybe even more exciting than our vaccine episode, because I don't think we can get vaccinated for everything. What's your take on this?
I think it's exciting too. I think there's already, I think, when you think about advanced imaging, better ways to see smaller things that might be going on in your body. And there's also a lot of research going on looking in blood, just a small amount of blood, for things like DNA and other things that could be coming out of tumors, that you could imagine a future where you give a blood sample and you get screened for the possibility of multiple different types of cancers that could then have you trigger some imaging. And I do think that we'll be able to diagnose cancer much earlier within relatively short order.
Maria, how early are we talking about with these diagnoses? When are people normally being diagnosed, and how much earlier should they be diagnosed?
You're asking me something that I really don't know very much about, and it depends on the tumor type, right? Something like ovarian cancer that my mom had, there's very, very poor early diagnostics for that. Some things like breast cancer, obviously can be diagnosed very early with screening, and colon cancer, right? So I think anything where there's already early screening protocols can already be treated, diagnosed and caught very early. Some of these other, like pancreatic cancer, some of these cancers are much harder to diagnose. So I think it's really dependent on the tumor type and what types of proteins they're making that are different from from normal proteins or signatures, so that you can distinguish them. Yeah, so going back to just in general cancer treatment, we've talked a lot about different modalities. You mentioned degraders, which I'd like to hear a little bit more about, and also to hear a little bit more about where you think the promise is. There's the immunology targets, there's molecular target. What are you excited about?
Yeah, as I said, this is such an exciting time for cancer research and also the development of new drugs, because we have such a toolbox of all the different modalities that we have at hand. And so, of course, we have antibodies that are specifically engineered to detect either growth factors or receptors on tumor cells. And that's how they were mostly used and developed. And, for example, anti-VEGF was a clear example of that, where we use antibodies to inhibit a growth factor that drives the formation of new blood vessels. But the technology for antibodies has expanded a lot. For example, with the development of antibody-drug conjugates, where you stick a drug target, either a cytotoxic payload, chemotherapy, or a more targeted drug, onto an antibody to deliver it specifically to the tumor cells, and trying to spare the other tissues. But also, for example, T-cell bispecifics, where you try to engage T-cells to tumor cells, where you have one part of the antibody that targets the T-cell, the other part of the antibody that targets the cancer cells, and you bring them together to activate in that way the T-cell and get anti-tumor responses. So there are many
forcing it. You're like, hey, immune system, over here.
Exactly.
Come look at this tumor.
Absolutely, that's exactly right. And so that just shows how many different possibilities there are using antibodies, and people have building and are still building on this every day. And on the other area, they had the more small molecules. You also have a lot of different possibilities. And the classical small molecules that, for example, covalently bind to drug targets, but also peptides, very small peptides that are very specific and disrupting, for example, the interactions of proteins, and degraders, as I said. So these also come in many different flavors, but the basic concept is that they bind to a target and they bind to something that is a part of the machinery within cells that is tasked with the turnover of proteins. Because also in normal cells you need to get rid of your damaged proteins, or when you have too many proteins, and they can put on a ubiquitin. That's actually a label to degrade a protein. You can imagine that if you bring one of those ubiquitin ligases to the target, stick them together again, that you can then degrade a molecule or a protein that is driving cancer, for example.
All right, so you're hacking a natural cellular process. The drug molecule isn't inhibiting the activity of the protein per se. It's just tagging it and saying, hey, let's get rid of this protein. It's like the trash compactor in the Death Star. Right? That's the way I always think about the...
No, that's a good way of putting it. It's just in the nature of cancer, which is it develops by an evolutionary process, and also in the resistance against therapies, you have this ongoing evolution that then selects for cells that are not sensitive to that particular intervention. And in often we use combinations to try and limit the chance that happens, because it's more difficult to develop resistance against 2 or 3 agents as compared to 1. And in particular in early lines of therapy, so that you would use in patients that have just been diagnosed with a disease, we often use combinations, and that's very important. And also puts specific emphasis on how well tolerated these drugs are, because you could imagine that if you combine drugs, that also comes with a cost of potential toxicity. So the bar for agents that you want to combine is really higher.
That makes a lot of sense.
Maria, I was curious about protein degradation. Louis was talking about that as a modality that is being used in oncology. Can you tell us a little bit more about how that works?
Yeah, so all of our cells are constantly making proteins, using them, and recycling them, right? So once a protein has sort of done its job, maybe it's got damaged, maybe we just don't need it anymore, the cell has a sort of trash compactor type of system that's just part of the normal functioning of the cell. And what protein degrader drugs do is hijack that. So it's essentially it's like it's big trash day. You would like tag the protein that's deranged in the cell and causing the cancer, and instead of just trying to inhibit its function or turn it off, you're actually just pulling it out of circulation and throwing it into the cell's trash can.
So Maria, do you have a favorite modality in oncology?
I do have a favorite modality, and I'm thinking about this from the patient perspective, and especially from my mom's journey, in that I really like monoclonal antibodies because of how much more tolerable they are to the patient to take. And so any modality that is easier for patients, that is not doesn't have the side effects of sort of traditional chemotherapy, really is, to me, a great breakthrough. I wanted to move on to talk a little bit about the role of AI and machine learning in the cancer field. What do you think about how that is changing the field of drug discovery in oncology?
I think AI and machine learning and the methods that relate to those will have such a far-reaching impact on everything we do, our everyday life, but certainly also the research we do. And I think you can really compare it with the use of computers or a calculator or something that would just completely transform how people do their jobs. And that's also certainly true for us.
Any specific examples of how you've used it in your own research?
Yeah, so one example that springs to mind from my own research in the past is that we have used it to identify molecular subgroups of cancer and to detect those in patient samples. So normally we did that by using gene expression data from a large cohort of patients, where we could classify them in particular subgroups of cancer patients that share, for example, particular features of the microenvironment or particular mutations in the cancer cells themselves. But it was always very hard to identify these patients in a clinical situation, because then we could not always do like a full genome transcriptome analysis or whole genome sequencing. So what we did at some point is to use image analysis software and an AI model to build a classifier that we could just use on a slide, a pathology slide of those patients, and we could then assign the subtype just based on that.
That's amazing. So the AI algorithm was able to look at a microscope slide, stained for the tumor or whatever, and say that I can tell by what I've learned from my black box of algorithm that tumor is going to be a class whatever tumor that expresses these genes and has this type of a microenvironment, and therefore will respond better to this treatment.
Yeah, that's exactly it. So something that took us like months to classify a patient to begin with, now we could do in just having a very simple
that's amazing.
Slide under a microscope.
And you don't need
a picture of it.
Sequence that patient's DNA at the cost of thousands of dollars or anything like that. You can just do it from what is a typical sample that would have been collected anyway.
And it really speeds up the research we could do, because then we could analyze much broader patient groups and relate it to clinical properties and response to particular drugs, etc. Just this one example, but
that's a great example.
There are many examples of this type. I think the impact on our work will be only bigger in the years to come, which is also good, because understanding cancer biology and utilizing those insights in discovering new drugs is a difficult task. We can use all the help we can get.
Yeah. And I think especially from a patient point of view, I mean, you don't have much time to do a lot of trial and error, right, with therapies. And that, my understanding, which is probably a little outdated, is that generally you have here's your first-line therapy, and if this fails, then you go to your second-line therapy. But do you imagine a world where you would be like, oh, no, we scanned your tumor, and it tells us this should be your first-line therapy, which is different from what it might be from somebody else's, because everybody's tumor is somewhat unique.
Yeah, I think, and sometimes we try to do this with very discrete factors. When a mutation is, for example, present, that then dictates if a patient is going to respond to drug A or to drug B, for example. But in many cases, we're also wrong. So to personalize the choice of therapies further, and to get to a learning healthcare system where we can also use maybe real-world data to further train and improve those algorithms to make better decisions going forward is I think, certainly a very exciting possibility.
It's very exciting. Any ethical considerations or health equity and inclusion considerations with that type of technology?
Yeah, I think, of course, those algorithms are in one way very smart, but you can also view them as very dumb, because they probably perform very well in spaces where they have been trained, but to move beyond that, I think is often challenging. So from the equity perspective, I think it's essential that we train those models going forward on as broad a populations as we can imagine, to make sure that they're also applicable across the board. Yeah, for other ethical considerations, of course, there's always some sort of the doom scenario that the doctor is not going to make the decision, but it's going to be the AI algorithm, and then who is to blame when the wrong drug is selected. I'm not so worried about that, to be honest. I think it will always be, at least for the foreseeable future, be just an additional tool that helps clinicians make decisions. But I think it will ultimately be an interaction from human to human that ultimately is the most important aspect of it.
What other theories in oncology are exciting to you right now? The vaccines, the cell therapies, some molecular targets that we have not yet been able to drug and we might be able to drug in the future. If you were to look forward 20 years, what do you think we'll be bringing to patients?
Yeah, I think all of the above, but... one thing where I think there is a lot of new discovery made is really to see how cancer cell intrinsic pathways that drive tumor development and progression and metastasis, how they interact with the microenvironment and the immune system. Because for quite a long time we've seen really these two things as separate. So either you can try and kill tumor cells by inhibiting a protein in the tumor cells that drives survival, for example, or you can activate the immune system, and then by that means kill tumor cells. But I think in the past years there is a lot of new discoveries and insights also in how tumor cell intrinsic pathways, how they regulate the immune system, and how they might suppress the immune system locally in the tumor. And I think when we understand that better, that would open up a whole potential class of targets, where you can find tumor cell intrinsic targets that manipulate then the tumor microenvironment and the immune system to achieve better therapies.
So bringing together sort of what have been the sort of two classical arms of cancer therapy into one. That sounds really interesting.
Yeah, because in my view, they are really two sides of the same coin.
There may be people who are listening to this podcast who have family members or who have cancer at the moment themselves. What would you say to people who are looking more near term? Like, I'm looking for treatments for my cancer now. Is it a personalized approach? Is that available to people?
Yeah, in many cases, personalized, or I would say therapies tailored to a group of patients, they are available and they are used. I think the more really personalized therapies for individual patients, that is something that is really in its infancy, right? And that's only, I think, what we would see in a horizon of 10 or 20 years from now. But I think on the nearer term in that area, and something that is, I think, very promising as well, is that you have these initiatives and studies to use cancer cells from patients in the laboratory, expand them, and then actually test in the laboratory, in culture dishes, which therapies are effective and which ones are not effective.
and in sufficient time to get that information back to the doctor to say, I took, Louis' cancer and I grew it up and it responded to this combination of therapies, and then you could get that information back to the physician in time to alter the treatment.
Yes,
wow.
that is possible, and clinical trials are run using this concept. Currently, this is only done in specialized centers, because you can imagine there's quite a lot of infrastructure that you need to be able to do this. But this, I think, could help us to understand better which patients respond to which therapies, and then to train, for example, these algorithms that might be in the future able to predict the sensitivity. But that is, I think, something that we might be seeing in some spaces in an earlier time frame.
So the degraders are super interesting. When do you think we might see degrader modalities actually making it to patients?
Yeah, they make it to patients as we speak. For example, in the hormone receptor positive breast cancer, there are many ongoing trials that use the concept of degrading the estrogen receptor. We know that the estrogen receptor is a good therapeutic target for these patients, but at some point these patients stop to respond to ER inhibition. And a new concept is then to degrade the estrogen receptor altogether. And that is really, showing very promising results in many different contexts and studies.
So we saved the tough question for last. Are we going to solve it? Will we have a future where nobody dies from cancer anymore?
That's a tough one. I think we're going to solve it in many cases, but I suspect not in all. Because ultimately, and I mentioned this already, like cancer is an evolutionary disease. And in many cases, I would suspect that evolution finds its way, right, around the therapies and around all the modalities that we talked about. But I think in the vast majority of cases, there must be a solution, and I think a cure could be reached.
That's very hopeful. Thank you so much, Louis, for your time. I really enjoyed our conversation.
Perfect.
Maria, this is such a great episode. I learned so much. I'm curious, what do you think the future state of oncology is from where you sit?
So this has been such an informative conversation, and it seems it's such an exciting time in oncology. There are so many breakthroughs and so many different types of pathways being tackled for different types of cancers that I really feel that we're starting to see cancer becoming more of a chronic disease, like even in cases where you can't say to somebody, oh, you're completely cured, but we have therapies that essentially can allow people to live much longer and much more normal lives after a cancer diagnosis. But on the flip side of that, it's still, a very, very difficult challenge, that there are still many cancers that are not treatable. And while we have really good ideas on the horizon of how these are going to be treated, it's not something I think we can get complacent about. I think it's a very exciting time where we're going to see a lot of breakthroughs in the future, but we really need to keep investing and working in the space.
From a data point of view, because of where you sit looking at lots of different things, is this a leapfrogging thing where one area makes these breakthrough advances and another, fails? And I guess my question is everything progressing together or is it iterative? What does the big picture look like from a data point of view?
So I think yes, both. It is the answer to your question. And I think one of the things perhaps you're alluding to, we're seeing is because of the ability to mine enormous quantities of data, we're able to have much more feedback from the clinic, what's happening when somebody is resistant to a drug, what's happening in their body, what's happening to the tumor, feeding that back to the scientists and iterating on why we fail, why we succeed, bringing all of that really rich data at the level of single cancer cells, at the level of the whole patient back to really inform future drug discovery. And that's our show. Thanks so much for listening. If you haven't already, rate our podcast wherever you listen. It will help new people to find us. And be sure to subscribe. If you have a question about the show, you can contact us at podcast@gene.com. That's GENE.com. And now for me, it's back to wrestling with data.